Okay, so now I will introduce our first speaker, Julia Freeman, who will be presenting her preliminary results on overlooked insects and an endangered endemic, Thrips as a potential pollinator for Hymenoxys texana. Okay, Julia, you can get started.
All right, thank you. I'll share my screen. All right, can you all hear and see everything okay?
Looks great.
Excellent. Okay, so yes, today, hello everybody. My name is Julia Freeman, and I am a master's student in the Milano Florist Lab at the University of Illinois. And today I'm going to be telling you about our ongoing research into the reproductive ecology and the pollinator communities of a rare and endangered endemic plant species, Hymenoxys texana, and specifically about the potential role of thrips as a pollinator for this species. And before I get into all of that, I want to start by saying thank you to our partners in Texas. These are the folks who are doing the on-the-ground conservation work for this endangered plant species. And all of the research that I'm about to tell you about today would have been so much more challenging without their very generous support and assistance. Okay, so starting just with a little overview of what I'll be speaking about. I'm going to be starting with giving some background and context about thrips as pollinators, and particularly as pollinators of plants in family Asteraceae. Then I will introduce you to our particular study system, our plant species, Hymenoxys texana, and our potential pollinator, Microcephalothrips abdominalis. And then I will go through, there are still ongoing methods to investigate this relationship. And then finally, I'll share some initial findings, all of which are aimed at trying to answer this question, are thrips pollinators of Hymenoxys texana? So starting with that background on the thrips. So for those who might be a little unfamiliar, thrips are tiny insects, mostly only a few millimeters in length. They have their own order, order Thysanoptera. And while there, of course, is a great deal of diversity within that order, they're pretty much all small and sort of long and torpedo shaped. Not all thrips have wings, but those that do have wings have these unique sort of feathery, bristled, hairy wings, which is where that name for their order comes from, fringed wing. Um, they're found around the world and they can be really devastating agricultural and horticultural pests. And so most of the published literature on thrips is focused on how to eliminate them as pests, which is, important and necessary work, but it has this really intense focus on them as pests has kind of obscured their other potential ecological roles, particularly as pollinators. So there have been three really exciting review papers that have come out just in the last 2 years about thrips pollination. And this is particularly notable because of how few published papers there are about thrips pollination at all. If you take a look at this bar graph, this is from Augustine et al., you'll see that there's fewer than 70 published papers about thrips as of 2024 at all. So the first paper documenting thrips as pollinators came out in 1914, and just the growth since then has been very, very slow. But at the same time, those roughly 70 or so papers have documented a great diversity of relationships between thrips and the plants they pollinate. So this figure is from another one of those review articles, Pop et al. 2025, and it shows a phylogeny of the different plant orders where we have either documentation of thrips pollination or very strong evidence that thrips pollination is likely to be taken place. And so Pop et al., they claim that because there have been relatively few studies on thrips, yet those studies that have been published just show this vast array of relationships, that it is likely that thrips are pollinators for even more plant taxa than are featured here, but just those relationships haven't been studied or published yet. And so I think that's something that's very exciting. Now, specifically going to family Asteraceae, let's take a look at Microcephalothrips abdominalis, the composite thrips. So as its common name suggests, this thrips species specializes on composite flower heads. And so not only is it feeding on pollen of plants of family Asteraceae, but it is also using the flower heads as brood sites. So it's laying its eggs within the involucral bracts and going through its whole life cycle within the flower head. And so Microcephalothrips abdominalis has been documented carrying pollen of more than 7 species of Asteraceae and is a documented pollinator, i.e., it actually increased reproductive output for one species, Meteoratum conizoides. And once again, this isn't saying that it's not pollinating those other species. It's just no one has taken their studies to this level yet, except for Gopinath in 1981. All right. And another species in family Asteraceae where Microcephalothrips abdominalis has been documented is Hymenoxys texana or Texas prairie dawn. So this is a gynomonoecious annual species, and it grows in patches of salty, sandy soil known as saline barrens. It is endemic to eastern Texas with populations found in 6 counties. So those are those counties filled in green over here, and with most known populations clustered around the Houston area. And most critically, it is both federally and state endangered. So the greatest threats to Hymenoxys texana are urban development, and occasionally mixing things up with some gas and oil pipeline development. I've taken all of these photos either within just a few miles of our research sites, or in the case of this one, actually at one of our research sites. That being said, there's no inherent reason why people have to be in conflict with Hymenoxys texana. And in fact, there's many individuals and organizations in Texas that are working very hard to conserve and protect this species. However, they're somewhat limited in their ability to do so by the lack of published peer-reviewed literature about Hymenoxys texana. And so the Milano Florist Lab is currently funded on a project to study the reproductive ecology of this species and fill in some of those knowledge gaps. So everything that we learn about through this grant can then be applied to on-the-ground conservation and even some reintroduction efforts for this species. And so today, of course, I'm focusing on the pollinator community aspect of it. So we've used a variety of different techniques to study the pollinator communities of Hymenoxys texana, and we have documented a diversity of insect visitors, including blister beetles like this Lyta bigutata, small lepidopterans such as skippers and sulfurs, and as well as small flies and bees, many of which have yet to be identified. However, all of these sort of quote unquote larger insects that have been visiting these flower heads have been very low in number and visiting only infrequently, with the exception of the thrips, which we had observed in abundance. And so we decided to follow where these observations were taking us and hypothesize that thrips are indeed pollinators of Hymenoxys texana. Now, to support this hypothesis, we are investigating 3 questions. So one, how prevalent are thrips on Hymenoxys texana flower heads? 2, do thrips carry Hymenoxys texana pollen on their bodies? And 3, does thrips visitation increase the reproductive output of Hymenoxys texana? Now, it's this third question that will actually tell us whether we can call thrips pollinators of Hymenoxys texana or not. However, it is also by far the most challenging question to answer. So answering questions 1 and 2 will help us build our case here. Okay, so getting into our methods. So all of the methods that I'm about to describe are taking place at 5 study sites located in Harris County, Texas, just northwest of Houston. Each site is a single saline barren, so this kind of brownish area in here, with a dense patch of Hymenoxys texana. So to answer our first question, how prevalent are thrips? We use two different strategies here. So firstly, we did presence-absence surveys of flower heads, where we lightly squeezed each flower head within our quadrants and then recorded thrips presence or absence. We paired that with a collection of a Hymenoxys texana flower head. So for each flower head, we sealed it in a plastic bag as quickly as possible, froze it, and then dissected it back at the lab. And then for each thrips that I found on those flower heads, I examined it under a microscope for pollen grains and counted the pollen grains on its body. And then finally, for our third question, do thrips increase seed set? So here we set up trials with 15 flower heads in each of 3 treatments at each site. So our first treatment is flower heads open to all insect visitation. Our second treatment is white mesh bags to exclude those larger insect visitors. However, the thrips can go right through the white mesh of these bags. And then 3, these red mesh bags to exclude larger insect visitors and deter thrips. There's some really neat research coming out of Japan on the pest side of things that actually using mesh that is the color red can be a deterrent to thrips. So that is what we have used here. Then once those flower heads develop into seed heads, we collect them, dissect them, and assess the seed set ratio, which is, of course, the number of plump fertile achenes divided by the total number of achenes for each seed head. And I should note here that we were only able to get seed set data from 3 out of our 5 sites because there are cattle at our sites, and they took a little bit too much of an interest in the bags at 2 of them. Okay, so on to results, and I should note here, all results are preliminary, so more information coming soon. Okay, so question number 1, what is the prevalence of thrips? So from our presence-absence surveys, we found that thrips are present on about 10 to 25% of flower heads, depending on site. And then when I dissected those flower heads, we found that there was a mean of about 3 thrips per flower head. And something fun here with the dissections is that it allows us to learn a little bit about the thrips behavior. So you'll notice that these thrips are fully encased in the florets and they're face down, which is how we would expect them to be if they were actively feeding on pollen within the floret. And we also observed thrips larvae and eggs as well as the adult thrips. And so this suggests that they are not just using Hymenoxys texana to feed, but that they're also using the flower heads as brood sites. Okay, then taking a closer look at those thrips, we found that they carry a mean of 9 pollen grains per thrips. And when looking at that pollen under a compound microscope, we can actually see some of the anatomy of the pollen grain and determine that it is indeed Hymenoxys texana pollen that they are carrying. Okay, and on to question 3. How do thrips impact reproductive output? If you take a look at this bar graph over here, so we have estimated mean seed set as a percentage on the y-axis, and then we have a bar for each of the 3 treatments. So the yellow bar here, we have flower heads open to all insect visitors, thrips and non-thrips alike. The gray bar here, these were our flower heads covered by the white mesh bags. So larger insect visitors could not access the flower heads, but the thrips could. And then finally, in our red bar over here, those are our red mesh bags, which exclude the larger insect visitors and deter the thrips. So I ran a 2-way ANOVA of treatment and site, followed by a Tukey's HSD test, and found strong evidence for a difference in seed set between those flower heads open to all insect visitors and the flower heads covered with the red mesh bag where the thrips were deterred. So to sum all that up on one slide, so we are indeed building a case for thrips as pollinators of Hymenoxys texana. We found that the thrips are present on flower heads, that they do carry Hymenoxys texana pollen, and crucially, that seed set decreases from almost 70% to about 47% when thrips are deterred from the flower heads. excuse me. So as I said before, all of this is still ongoing. So we did another round of those insect exclusion trials in the field this spring. And we also set up a very similar experiment in the greenhouse inside of these bug dorms, where we can test out our mesh bags in the absence of cattle and have a little more control over the situation. So those are all still in progress, and I'm excited to see how adding in that data will affect our final results. And so I will be at the Botany 2026 conference in August. I'll be presenting a poster there. And so if you'd like to talk more about thrips pollination or about rare and endangered species and family Asteraceae, I would love to speak with you more about it then. Well, thank you very much.
Thank you, Julia. Yeah, that was a great talk. So, do people have any questions?
Yes, Miranda.
Oh,
Oh,
I was applauding, but I actually do have
thank you.
a question. I thought it was interesting how your can you go back to the seed set slide where you have the 3 bars?
Oh, yes.
Yeah, do you I, sorry, my brain is a little bit slow right now. What I thought was interesting is that it looks like the thrips are obviously important, but the other pollinators are as well. Do you have any, like, do they interact with each other at all? Um, do they, do you think one is more important than the other? I mean, I totally agree that it looks like thrips are definitely pollinating, but I'm just curious if you have any more to say from your observations or whatever, about that particular result.
Yeah, definitely. So there's a lot going on here for sure. And we've also been doing breeding system trials on this species. And so from there, we've been getting some preliminary evidence that this species can self-pollinate and self-fertilize as well. So there is some of that happening too. So even if no insect pollinator can access it, we're still seeing like around 30% seed set. So there's the selfing that's happening, there's the thrips, and then yeah, we have certainly seen other insect visitors as well, the small bees and flies, the small lepidopterans, the blister beetles, which are like really fantastic and showy. We've actually only seen those at one site, so that's kind of interesting. Um, I think the thrips are notable because they've been so consistent. Our other insect visitors, depending on the weather, depending on the time of day, depending on which year, it 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 can really vary, like who is showing up and how many of them are showing up versus the thrips seem more steady. So yeah, I'm still working through a lot of our insect visitor data, so I can't give like a strong quantitative response about how much different groups are contributing. Um, but yeah, there's definitely more going on than just the thrips.
Very cool. Yeah. There always is. It's never quite that simple, right? So it's super
For
cool to see
sure.
all these other, like the whole community. Yeah, very cool. Very cool talk. Thank you so much.
I was wondering if, do you think that thrips are likely to be important pollinators for many different Asteraceae? Or do you think there's something special about your species that makes them especially good?
Sure. Let's see. So those three review papers that I mentioned earlier, the different authors sort of have conflicting views on whether there is like a unified syndrome for thrips or not. And it does seem really variable. One thing that a lot of flowers that are attractive to thrips have in common is having like really small, tight crevices that thrips can squeeze into and kind of protect themselves from the elements or predators or other insects that might be accessing the flower. So I mean Hymenoxys texana certainly has that with its tiny little florets, but so do so many other flowers and family Asteraceae. So um, yeah, based on my reading and our observations, I will say since I just became aware of thrips, I feel like I see them everywhere now. Um, thrips are probably doing a lot, and we just don't know yet.
Very nice. Okay. Well, thank you. Any further questions for Julia?
I have a quick question, Julia. You
Yes,
mentioned that the species is Gynum monoecious.
yes.
Have you been able to see, a seed, an achene from the female ray flowers? I'm curious.
Oh, great question. Let's see. Typically, when dissecting, I haven't been trying to keep the ray florets separate from the disc florets. And not all the individuals have the ray florets. So I'm not sure. We have, however, yeah, we have dissected seed heads where the seed set has been 100%. So every single seed that we can tease apart there has been fully plump and developed. Um, but I don't know if those seed heads had ray florets when they were flowering. So yeah, that's something. There's more seed head dissection to be done. So that's certainly something I can pay attention to in the next round.
Okay, well, thank you so much.
Okay, very nice. Thank you. So our next talk is from Miranda Sinnott-Armstrong on defense traits across leaf age in thistles. Cardueae.
All right. Guess we will try this and that. How's that look?
Looks very good.
Oh, y'all disappeared though. So shout if you want to ask a question or anything like that.
Sounds good.
Yeah, so as Abby said, I'm going to talk about defense traits and how they vary across leaf age in the thistles. Sorry for my voice. I'm getting over a horrible cold or I don't even know, something like that. So I hope it doesn't bother you too much. But yeah, let's just get right into it. So a lot of plant morphological diversity it's often been attributed to interactions with herbivores. And this idea goes back to Ehrlich and Raven, where they essentially propose that there's this kind of coevolutionary arms race between plants and herbivores that is kind of continually generating mutations and adaptations, leading to a lot of this plant morphological diversity. So this plot right here is showing an example. On the x-axis, we have evolutionary time. And then on the y-axis, we have the amount of herbivore-caused damage. And so the idea is you have a plant, it has a lot of herbivore damage, suddenly it acquires a mutation that provides it some protection, and the amount of herbivore damage goes down until you reach some sort of new adaptive zone, at which point potentially an insect or whatever herbivore gets another mutation, which then allows it to overcome the defense that the plant evolved and increase the amount of damage again. And so you can end up with this kind of cycle of plants evolving defenses, insects evolving kind of ways to overcome those defenses, and that this cycle can then generate a lot of diversity. And of course, some of this idea is that maybe you bounce back and forth between extremes, and maybe eventually you evolve some sort of static or closer to static balance between plant defenses and insect or other herbivore like attacks. So it's thought that this kind of a process is one of the things that generates a lot of plant morphological diversity. And indeed, we do see that plants have evolved a wide array of defenses. So there are a few that, were probably quite common or that we're familiar with because they're common. Things like trichomes, so hairs on the surface of leaves, but they can also be on stems and other tissues. There's the toughness of the leaf, leaves can be tough for different reasons. They can be really thick, they can have lignin, but essentially kind of being a tougher leaf means that it's going to be harder for especially small herbivores to actually eat the leaf. Spines, of course, are something that we are probably all familiar with. Maybe you have stepped on some plants that have a lot of spines and it is not a fun experience. Maybe you've tried to eat raspberries or blackberries and gotten yourself really, attacked by I use here spines loosely in the context of like spines, thorns, prickles, whatever. But these, sharp, pointy things, right, that help to defend the plant against herbivores. So those are a couple examples of physical or structural defenses. But plants also often produce sometimes a really astonishing diversity of chemical defenses. So things like alkaloids, glucosinolates are some common types of plant chemical defenses. But the reality is that there's just a huge diversity out there. Some lineages will kind of have a lot of one particular type of chemical defense. Other lineages might use a completely different set of chemicals. Some lineages might use a lot of different chemicals instead of just one. There's just a huge amount of variation in the chemical defenses that plants produce, in addition to and on top of these physical structural defenses that are things like trichomes, tough leaves, and spines. We've often thought about this kind of plant diversity in defenses in the context of what we call defense syndromes or defense strategies. There's actually a few different names for this idea, but fundamentally the idea is that there's kind of combinations of traits that tend to go together because collectively they provide a pretty good defense against herbivory or whatever the challenge might be. In this case, we're talking, of course, about defense syndromes, so provide protection against herbivory. And one of the first descriptions of this in the context of plant defenses was Agrawal and Fishbane, who found 3 different defense syndromes, if you will, associated with fast growth and just not having very many defenses at all. The idea here is that the plant can just basically outgrow its herbivores. So even if it loses a lot of tissue to those herbivores, it just keeps on growing, right? And it just grows and grows and grows. And, it still functions. It's fine. Like it survives enough to reproduce. But it's kind of trying to escape that herbivory by just growing so quickly that it doesn't really care if it loses a bit of tissue here or there. Then the second big syndrome that they found is associated with slower growth, but chemical defenses. And then a third would be slower growth and structural or physical defenses. And so, this idea about syndromes is a very attractive idea. And people are often looking for syndromes in all sorts of different contexts, whether that's defenses, pollination, seed dispersal, et cetera, et cetera. We're often looking for these kinds of patterns. And the reality is that when people have started to look for these syndromes in other groups of plants, they sometimes find the same syndromes, but they sometimes find kind of completely different combinations of traits that tend to co-occur. So one example here, we have, again, kind of clusters of traits that co-occur across different species, but those clusters are not the same suites of traits that Agrawal and Fishbane found. So this red group of species here has faster growth and low defense, which is more or less that first syndrome from Agrawal and Fishbane. But the blue species are slow growth and high defense, but their defenses are specifically a combination of chemical and physical defenses. So they have high flavonoids and low SLA. SLA being specific leaf area. So if you have a low SLA, you have kind of more leaf mass packed into a smaller area, which basically means you have a thicker, tougher leaf. If you have a high SLA, then that mass is spread out over a larger area, and so you have a thinner, more papery leaf. And so high flavonoids, low SLA is a combination of chemical and physical defenses. And then we have this green group here that has, again, slow growth and fairly high defenses, but again, it's a combination of physical and chemical defenses. So these plants have high lignin and high tannins, which again, physical defense, chemical defense, which doesn't exactly align with the types of syndromes that Agrawal and Fishbane proposed. Now, I only want to belabor this point because I think when we think about syndromes, we're often kind of trying to look for some sort of universal suite of traits that kind of co-occurs across all the groups of plants that we can find it in. But the reality when you look at syndromes is that it tends to be much more like this, where, one group of plants, you might find kind of consistent patterns. And then another group of plants, you might also find consistent patterns, but they may or may not be the same patterns across all of these different groups. And so that complicates how we want to think about the evolution of these traits, these so-called syndromes, because we want to make sure that we're really looking at traits that are relevant for each particular group that we are studying. The other thing that I want to kind of bring into this whole picture is that young and old leaves can actually look very, very different. This this is like an artistic rendition of an herbarium specimen. But what we can see, even from this kind of artistic drawing, is that we have kind of larger, older leaves that have really big spines, and then we have tiny leaves with smaller spines in the young leaves. You'll find out soon, actually, that's not very accurate, but whatever. But the point is that you see this ontogenetic variation. And that variation across the development of the plant and across the development of the leaf doesn't necessarily match the syndromes that we see in maturity. And instead, we may see kind of different syndromes occurring at different ontogenetic stages. And just to clarify, when I say ontogenetic, I mean developmental stages. So we're talking about old leaves, intermediate age leaves, young leaves. You can also apply that term to the whole plant. I'm just talking about leaves in this context. But the point is that we might very well see that there are different syndromes, different combinations of traits that occur at different ontogenetic stages. And in the context of herbivory, part of the reason that we might expect that is because young leaves and old leaves are often susceptible to different types of herbivores. So there's a lot of variation here, both spatially, geographically, phylogenetically, but in general, we tend to think that young ontogenetic stages, so young leaves, are usually more susceptible to insect herbivores, while older stages tend to be more attacked by mammalian herbivores. And those two different types of herbivores create different selective pressures on leaf traits. And as a consequence, plants evolve different suites of traits to protect against those different herbivores. And so it maybe isn't surprising that we actually do see different combinations of traits and kind of leaves that look different at different ages, perhaps because they have evolved to protect themselves against different herbivores that they're more susceptible to at those different times in their lives. So one kind of caveat to all of this work is that there's been a lot of work on plant defenses, but a lot of it has focused primarily on tropical species, on woody species, and honestly, a lot of the time, it's like one chemical trait or one structural physical trait. Of course, that is not universal. There are studies that go outside of these kind of tendencies. But when you look at a lot of this literature, it often is focusing on this kind of more limited subset of plant diversity. And so what we wanted to essentially know is, are herbaceous species following the same patterns as some of these tropical woody species? And also, when we look at kind of a broader suite of traits and we don't focus in on only one chemical trait or only one structural trait, what kinds of patterns do we see in how defense traits and defense syndromes vary across species and across ontogeny? So we wouldn't be here today if we weren't all interested in composity. Composity. I can't even say that word. Anyway, we wouldn't all be here if we weren't interested in the same groups of plants. And so today I'm going to talk about the thistles, which is a mostly herbaceous lineage in the Cardueae. You guys are probably all familiar to some extent, at least, with the thistles. One example is Cirtium vulgare, the bull thistle. And that plant is nasty. Like, I don't know if you've ever encountered it in the wild, but like, you do not want to touch it, right? Because it is covered in these enormous spines, and they do really hurt if you step on them. So I don't recommend that. But the thistles vary enormously in these defense traits. Now, spines are just one of the kind of most visibly obvious traits that we can see. But even in just the spines, we have species like Cirtium vulgare. We have species which have spines, but they're not quite as big or as aggressive. We have species that have kind of like pathetic spines, but they are sort of there. And then we have some species that have essentially no evidence of physical defenses at all. And so across the Cardueae, we have this enormous variation in physical defenses, and as you will find out, also in chemical defenses, which makes them a really interesting group of plants to study the evolution and diversity of defense traits and syndromes, both across species and across ontogeny. The other thing that I want to quickly add is that many of these species are quite cosmopolitan. Many are invaders and really nasty invaders. And some of that success in invading may have to do with their ability to resist herbivory. And so the kind of defense trait, defense syndrome side of this is not just of interest from kind of how they evolve, but also for potentially understanding why these species are so invasive. So essentially what we did is we went out and we quantified a whole bunch of defense traits, both structural or physical, as well as chemical. We looked at trichome density, we looked at specific leaf area, we looked at a number of different traits related to spines. And then for chemical traits, we used an approach called untargeted metabolomics to help identify molecules belonging to common defensive pathways. So pathways like alkaloids, flavonoids, terpenoids, and phenols. So how do leaf defenses vary across leaf age in the thistles? To kind of get back to this idea about ontogeny, we have leaves of different ages as you go up the stem of the thistle, which, to be very fair, means that they also differ in position, not just in leaf age, but they differ in kind of both position and leaf age as you go up the stem. So we have older leaves, which tend to be bigger on the bottom. You have intermediate leaves, and then you have young leaves, oftentimes kind of right below the reproductive material, the head of the thistle. This is an example of what leaves of those 3 different ages look like in one of our samples, one of our individual collections. And you can see that in some ways they look really similar. They're kind of lobed, they have spines, but in other ways they look very different. For example, the young leaves are much smaller than the old leaves. And we will get back to this quite a bit, but the length of the spines kind of more or less looks like it's the same across these 3 leaves. But in the young leaves, that means that those spines are like a huge proportion of the leaf size, whereas in the older leaves, it's a much smaller proportion of the leaf size. So that's just an example of how these leaves vary across ontogeny. Okay, so when we take all of this data, I'm sorry, I should back up 1 second. We got collections from 7 different species and multiple populations and multiple individuals for most of those species. Some of them we could really only find like 1 or 2 plants, but for most of them we have multiple populations, multiple individuals, and all of that is kind of bundled into the single analyses that I'm going to show you now. So when we look at structural traits and how they vary across leaf age, we see a couple of patterns. So here, we've basically taken all of our measurements for these different traits and then standardized them so that we can put them all onto a single graph and kind of look at the effect, if you will, or sort of how much these different traits are varying within each other, and then compare that to the other traits. So because these are all like totally different units, we wanted to put them on a single plot. That's why it says values, because it's this kind of like standardized value for each individual trait. And what you probably see immediately is that a lot of these lines look like they're maybe going up or kind of flat, but then we have this relative spine length, which is dropping precipitously. That's something that I just mentioned, where the young leaves have spines that are kind of more or less the same size as the old leaf spines. But because the leaf is so much smaller, those young leaves are just like all spine. They have like a little bit of leaf and then a ton of spine. Whereas the old leaves tend to have a lot more individual spines, but they also have a lot more like actual leaf tissue that those spines are then protecting. The kind of yellowy star lines there is the density of trichomes. It tends to decline slightly on average across species. The reality is that in some species, that decline is very strong, and in other species, it kind of more or less stays flat. But when you average essentially across all of these species, you see that trichome density on average declines. And then aside from this relative spine length, we tend to see that other spine traits are increasing. So the length of the longest spines gets a little bit bigger, flat or a little bit bigger, and the spinosity, which is kind of like the density of spines around the margins of the leaf, again, gets a little bit bigger, but it's not like a huge, I don't know, it's a pattern, right? It's there. It's not like an enormous pattern, but we do see like a few more spines, higher density of spines in these older leaves. For chemical traits, we use this untargeted metabolomics approach, and then we basically assigned the molecules that we found through that method to different pathways. And then we picked out the main kind of chemical defense pathways that, there were others that were in this data set, but we kind of picked the main ones where we had a lot of different molecules. And so what we see here, each of these lines is like the number of individual alkaloids or of flavonoids, phenolic acids, or terpenoids. The number of chemical features is kind of like the overall number of molecules that we found. And then the chemical functional diversity is essentially when we're assigning those different molecules to different pathways and kind of identifying them, we know that some of those are really closely related, like flavonoids are all really closely related. They're part of the same pathway. But when we kind of look across the whole diversity, we're sort of like, oh, well, maybe this one has a couple of flavonoids and a couple of alkaloids, whereas that one just has a lot of flavonoids. There's greater chemical functional diversity when you can look across these different pathways. It's almost like a chemical phylogenetic-ish, not really phylogenetic, obviously, but kind of like trying to capture that idea that some of these molecules are related and are part of the same pathways. And if you have molecules from lots of different pathways, that's more functionally diverse than if you have just a lot of molecules from a single pathway. Hopefully that more or less makes sense. The main patterns that we see here is that the chemical functional diversity actually increases across leaf age. So that's that yellow star line in the middle that goes up, which is interesting because it suggests that potentially these older leaves are pruning down like unnecessary molecules and only keeping like the really important molecules, which means that they just have like a greater functional diversity because they don't have a lot of the like extra molecules that they're not really using anymore. And or it means that they may be adding on new pathways, like entire new pathways, whereas the younger leaves have lower functional diversity, but they tend to have a lot more individual molecules of these different major defensive pathways. So there's like a lot more flavonoids in the young leaves than the old leaves as individual molecules. So we see these kind of interesting patterns about chemical traits where the number is declining, but the diversity is actually increasing across leaf age. And this is roughly in agreement with the idea that chemical defenses in early development, with the idea that chemical defenses are stronger in early development, while structural or physical defenses tend to be stronger in later development. And that's that same pattern that I had already talked about earlier. Well, excuse me, when we think about this in the kind of context of syndromes, what we see is that there's this kind of shift in traits across developmental stages across species. So here I'm showing a PCA of all of those traits combined. And of course, we have young leaves in the purple and then kind of intermediate leaves in the tealy color and old leaves in yellow. And you can see that they're each overlapping, but there is this kind of shift in trait space as you go from young to old leaves. And when we parse that out by species, what we tend to see is that the species kind of all shift in the same direction, but their starting points and their ending points are different. So if we look at the blue convex hull here, which is from Sertia marvense, excuse me. If we look at this blue convex hull from Sertia marvense, you can see it goes from kind of high in PC2 down to the middle of PC2 in old leaves. Sertium vulgare, which is this yellow one at the bottom, starts in the middle for PC2, but then goes really low in PC2, right? So they're kind of showing that same pattern, the same shift. It's just what exact traits are involved kind of varies across these different species. And so it's interesting because it suggests that there are these kind of general patterns about how traits differ across ontogeny, but it is also kind of species specific which traits exactly are involved in those shifts, which I think is pretty cool because, you know, it suggests that like maybe there is some more general selective pressure by, let's say, insect herbivores and young leaves, but at the same time, that's like written on top of the differences across species, right? So anyway, I think that's a really interesting pattern. The other thing that I wanted to point out is that roughly speaking, what we see is that young leaves tend to be a little bit more similar across these species. Of course, you know, they do occupy slightly different regions of PC space, but they tend to be kind of fairly similar. But then old leaves is where we start to really see differentiation. We see like Sertium vulgare is in a totally different region of PC space than Sertia marvense in the old leaves. But in the young leaves, they're like close, maybe partly overlapping. So we start to see these young leaves have kind of more similar suites of traits. And then there's a lot more differentiation in the older leaves as this, you know, ontogenetic process unfolds. And then finally, I wanted to talk about some more macroevolutionary patterns. We don't have a ton of data here. We have 21 species, and I think it was 34 samples total. We have here on the left a phylogeny of the Cardueae with species for which we have data on spininess or non-spininess. You can see that the dark green, which is spiny, has evolved multiple times across the group, although there are, some places where spininess is like clearly a very strongly inherited trait. But when we then sample our 21 species across that phylogeny, what we see is that actually spiny and non-spiny taxa do slightly at least have different chemical profiles. They overlap. It's not kind of like a perfect differentiation, but this NMDS plot in the middle is an NMDS of the untargeted metabolomics data that we have from herbarium specimens for these species. And so essentially what we see is that they are occupying slightly different regions of trait space. We don't have a ton of data here. I would love to get more. If somebody wanted to go out and sample a lot more taxa, that would be amazing. So it's really at this point kind of just suggestive that perhaps spiny and non-spiny taxa have slightly different chemical profiles. All right. So just to summarize, I actually kind of skipped some of this in the talk, but we see that there's variation in these defense traits at all biological levels. Across leaf age, we talked about quite a bit, but we also have analyses that talk about individuals and populations. And then, of course, across species, we also see that there's a lot of variation in defense traits. And so really kind of at all levels of biological organization here, we see that there's variation in both chemical and physical defense traits in the Cardueae. Roughly speaking, the patterns that we find do comport with this expected. Hold on, guys, sorry. It roughly comports with this idea of an expected transition from mostly insect to mammalian herbivores. The reason is because insects are often especially susceptible to chemical defenses, which are stronger in the immature leaves. We also see that trichomes can be particularly protective against insects, which again, more common in the young immature leaves. And so we see that these kind of traits in the younger leaves are often the traits that we see in conjunction with insect herbivory. And then the older leaves with their really big spines and often slightly thicker leaves, those are traits that tend to be more protective against mammals. So roughly speaking, the patterns that we see here comport with this idea of an expected transition from mostly insect to mostly mammalian herbivory. And I will say, we did not even attempt to monitor that or count that or observe this pattern. So this isn't purely like a correlation of traits and what we might expect to see. And then one of the things that I personally find the most interesting is that we do not actually see that these traits form clearly distinct syndromes. Instead, we see that there's this continuum from fairly undefended plants to very heavily defended plants, where that axis of variation is just shifted across species and across age. And so in the young leaves, we have this continuum. In the old leaves, we have the same continuum, but which traits are involved and where exactly those species are in that continuum varies across these ontogenetic stages. And so there's this really interesting continuum that we observe as opposed to these distinct syndromes, which quite frankly, people are always looking for distinct syndromes. And so anyway, I think it's very interesting that we see this kind of shift in which traits are involved, but overall the same pattern of undefended to heavily defended as opposed to kind of distinct clusters of traits. And then finally, across Cardueae, we see that spininess is associated with a distinct chemical signature compared to non-spiny taxa. Again, that is kind of a preliminary result. It would be great to get a lot more data to really dig into that, but it's certainly an interesting observation that might very well, yield some interesting results in the future. Okay, and so with that, I will take any questions. I too am going to botany. I hope to see you there. I will not be talking about this project, but I will be talking about the kind of next project in the realm of thistle defenses, which will be talking about thistle head fauna diversity. So please come if you are interested. And then I also wanted to say that I'm going to be joining the Cleveland Museum of Natural History in July as curator of botany. And so if you or anyone you know is looking for postdocs and wants to write fellowships or grant applications, although we all know how that's going right now, but wants to, potentially do a postdoc on any of these topics about defenses in thistles or other plants, please do let me know. And yeah, with that, I will take questions. I think I'm going to have to stop my sharing because I can't actually see anybody. So
Thank you, Miranda. That was really interesting.
I'm going to put this up there. Thank you.
So do people have questions? Marissa?
Yeah, I was curious about the trichome density over age. This is something that I've kind of wondered about with my own plants, where a lot of the really young leaves will be kind of like silvery or canescent, but then some of the older leaves that have had time to kind of broaden out and kind of reach their full size, they're often lower in density, but I'm not sure if the plants or leaves are making like relatively stable amounts of hairs, and thus the density changes like with age because the leaves are expanding without making more hairs. Is that something you've kind of observed or noticed?
So we did not like quantify that exactly, but there is some literature about that very question of like, is the plant producing new hairs over development, or is it just like it makes them all at the beginning, so they're all packed into a smaller area. Then as that leaf expands, they just become less dense. Um, I think mostly in this case, it probably is more a matter of as that leaf expansion is happening, maybe that's kind of leading to the lower density. But there are some papers that show that like they do make new trichomes, or at least they can make new trichomes. So it may be something that's very like system specific, whether or not your plants in particular make new ones or just put them out at the very beginning. The other thing that is kind of like important, I guess you could say, about these plants is that they do have this variation in ontogeny with like the old leaves and the young leaves, but also that is associated with their position on the stem and how close they are to reproductive material. And I don't think that these young leaves at the top are likely to like ever become the same size as the old leaves. I think they're kind of like, that's just that's their morphology. That's their like final morphology. And so, that would suggest that perhaps it's more of a like adaptive thing as opposed to just like a leaf expansion thing, that the younger and higher position leaves have this different density of trichomes. But yeah, we actually debated about that very question a long time, and we're like, what do we do about this, and like what kind of patterns are happening in these plants versus other plants? And I think it's just something that we haven't studied enough to have a definitive answer, and it probably does vary system by system. But it's a really good question.
I have a question. Um, so for the chemical analysis, you did it using intact leaves?
Uh, I mean, yes, we had to pick them off of the plant, but we tried to choose leaves that had not been eaten by an herbivore, if that's what you're asking.
Yes. So then if they are, and I don't know if you did it, how that chemical composition changes among the different leaf stages.
Yeah, we have not done that, but I would love to do that. That would be really cool. And especially if we could identify leaves that have been predated upon, I guess, by insects versus by mammals, and see how that varies across the ontogenetic stages. Yeah, I think that would be really cool. We did not do that. I wish we had. I wish we could. Hopefully we will. Yeah.
And what about the ones that are associated with the inflorescence, with the flowers?
Yeah, so we only looked at leaves that were high up on the stem, close to the inflorescence, but not actually like really associated directly with that inflorescence. We do have a little bit of data about the filaries, but yeah, we didn't look at that part of it as much, but that would also be really cool because I bet those structures are doing something totally different than like all the other things. So yeah, there's definitely a lot of other things that we could do with this, which would be really cool. So
Okay, awesome. Thank you.
yeah.
Hey, well, thank you very much. That was very interesting.
Thanks, everyone.
So now I will introduce our last speaker, Thaabiet Parker. I'm sorry, I am sure I messed up your first name, who will talk to us about drivers of clinal morphotype persistence despite widespread gene flow in the African daisy species complex Dimorphotheca pluvialis-sinuata. Let's see, are you able to share your screen? No, we cannot hear you, unfortunately. We cannot hear you and we can't see you or see your screen. You don't look like you're muted, but something is not working. No problem.
Hello, can you hear me now?
Yes, we can hear you and see you.
I'm so sorry about that. I'm not sure
No,
what that
no problem.
just cutting me off. Sorry, everyone.
And I
Let me.
will. Okay, you should be a co-host, so you should be able to share.
Yeah. All right. Can everyone see?
Yes, we can.
Great. Okay, I
Perfect.
can't see anything. So cool.
Okay.
Okay, sorry about that, guys, and for the delay. So hello, everyone, and good day and good morning to the US colleagues, and good evening to other people around the world. It's 9 p.m. where I am at the moment. My name is Thaabiet Parker, and I'm a taxonomist at the South African National Biodiversity Institute, where I currently work as the curator of the Composite collection in the Compton Herbarium in Cape Town. So it's my pleasure and my privilege to present in this forum. And today I'm going to speak to you about a chapter of my PhD thesis, which I just passed this year, thankfully, entitled "Drivers of Morphotype Persistence Despite Gene Flow in the Dimorphotheca pluvialis-sinuata Complex." I've previously spoken about aspects of my thesis in other conferences, but this chapter I haven't presented. And I sort of snuck it into this ecological focused TICA session. So forgive me for that. But I thought it's quite an interesting topic to bring because it deals with many of the problems that systematists deal with in species limitation, and particularly composite systematists deal with in recently diverged systems. So right into the conceptual background of this study, if we think about problems with species delimitation in the modern era, the unified species concept has become the consensus. So we think of species as independently evolving lineages, and this is sort of a broad definition, but one which has an ontological basis. But speciation often occurs with gene flow, and this can cause complications for identifying independent lineages. In particular, there, for example, two common scenarios are hybrid speciation and integration, where lineages which are presumed to be independently evolving exchange genes. And these can result in population networks which are represented as paraphyletic phylogenies in downstream inference. And so under these situations, monophyly is essentially violated. And monophyly is a criteria that's often used to delimit species. And morphology becomes blurred and essentially clinal zones form. So here in this schematic, you can see a transition zone between a species which is white, a bird which is white, and a species of bird which is black. And in the transition zone, the morphology becomes gray, and presumably the genetics also forms a cline like this in the hybrid zone. So just a brief introduction to the study system I was working with in my PhD, which will form the basis of this presentation. It's a complex of two annual species, Dimorphotheca pluvialis and sinuata. They are endemic to South Africa. And in the current taxonomy, they're separated by orange versus white ray color. But obviously, that's a very simple morphological difference. And in fact, when we looked at field observations, we found there are as many as 19 floral morphotypes present in the complex. And I did an integrative taxonomic study, including genetics, morphology, and crossing experiments. And what I found was consistent evidence for three species, which were reproductively isolated, monophyletic, and also morphologically diagnosable. But as you can see by this graphical abstract schematic, some of the species contain multiple morphotypes, so a large amount of morphological diversity. And these morphotypes are parapatric and morphologically diagnosable, but completely not reproductively isolated. And so I elected to raise them to subspecies level on this grounds. But this doesn't sort of answer the question of why do these morphotypes actually persist in the landscape in such a consistently diagnosable morphological fashion. There's consistent gene flow between them, and they are not reproductively isolated at all. So I began the study by setting up three different hypotheses. And the first one, which is probably the one that would first spring to mind, is the possibility of divergent selection. So when you have rapid range expansion with gene flow, you can then get selective pressure, which fixes traits along this ecological gradient, resulting in divergence through reproductive isolation. So in the case of this system, I thought pollinators were the most likely based on previous studies, and also based on the fact that all the variation was floral, so most likely selected on by pollinators. And then a second hypothesis would be allele surfing, which is an extension of genetic drift. And this process basically dictates that sequential range expansion with interrupted gene flow will result in traits being fixed in edge populations, and specifically traits associated with reproductive isolation. And this can result in these morphotypes evolving. And the last hypothesis is IBD, which is isolation by distance. And this is not a speciation process, but effectively genetic drift, which results in transient variation in these traits due to spatially constrained dispersal. So you get somewhat subtle change in genetics and in morphology over the distribution, just as a virtue of how far populations are from each other, but they're not related to reproductive isolation. So the focal study transect in the system that I looked at for this study was a transect of 4 morphotypes. I called them by quite rudimentary morphotype names. They're now going to get subspecies names, of course. But these morphotypes were separated by variation in bandage on the ray lamina, and also by variation on the inner disc corollas. They have different types of appendages on the disc corolla lobes. And as you can see, they have a parapatric distribution along this north-south transect in the Olifants River Valley in South Africa. So for the study design, I essentially took multiple samples from each population that you see here on this transect. And that was genetics, so GBS data, next-gen sequencing data, morphology in the form of disc and multispectral photography color traits, and also 2 selective regimes, the pollinator frequencies that I've spoken about before, and soil variables. So the first step for the analysis was to basically identify the pure morphotype endpoints of the transition zones between these morphotypes. And what I did was an SNMF of genetic data. So that's basically a population genetic admixture analysis. And I first did an SNMF on all the samples to kind of find the points where the gene flow patterns changed, where there were less admixture with the other end of the transects. And then I did a subsequent analysis where I split each of the transitions zones into separate and ran SNMF on each separate transition zones. And these both to collaborate the pure endpoints in terms of the genetic, the gene pool assignment. And you can see right off the bat that actually the Cardueae citrus dial transition zone is actually poorly genetically differentiated. The SNMF of all the samples couldn't find 2 different gene pools in the admixture analysis. And even in the specific transition-specific analysis, there is admixture in the pure morphotypes on the end of the transition zone. And these morphological differences these pure morphotypes were corroborated with PCA to show that they were actually morphologically different as well. So I then proceeded with modeling Clinal variation in genetics morphology and the selective regime using the HSAR package in R. And in terms of genetics, what I modeled was outlier allele frequency. So this was SNPs, single nucleotide polymorphisms, that were fixed on either end of the transition zone. So this was supposed to be sort of analogous to speciation genes. So which genes are associated with each morphotype. And then I also modeled the Clinal variation in SNMF coefficient. So that was basically analogous to the whole genome signal. Yeah. And then in terms of the morphology and the soil and the pollinator frequency, selective regimes was a bit more difficult, but basically because these were multivariate, I had to transform these variables into single variables. And what I did was I used PCA and used the transition-specific endpoints as the 0 and 1 on the PCA effectively. And so I then modeled each individual along the transition zone as representative of how close to each of the endpoints they were in terms of morphology and soil. So the expected clinal patterns that we would see from these analyses is in divergent selection and allele surfing, you'd expect the genome-wide signal and outlier allele frequency to be a steep sigmoidal shift in the transition zone, highlighted in yellow here. And you'd expect phenotype and as well to have a steep shift. But importantly, for divergent selection, you obviously expect the selective regime to be strongly correlated with the shifts in genetics and phenotype. Whereas with allele surfing, you'd expect it to either there to be no pattern in selective regime or it to be uncoupled from the other shifts. Whereas with IBD, you'd expect shifts, but the shifts to be much more subtle, so a less steep climb in genetics and phenotype. And again, you'd expect no pattern in selective regime. So into the results of the analysis, if we look cline by cline, this is the peach-K tricolor cline, and each of these represent a cline or shift in genotype, phenotype, and selective regime. Here, where the x-axis is the distance on the northernmost point on that specific transition zone. And what you can see is there's a set of outlier SNPs which shift steeply along the transition zone. And as well, genome-wide signal also shifts steeply along the transition zone. What I didn't mention also was that I split the morphological data set into two as a function of some sites not having multispectral data. And as you can see, these shifts were more linear and less, and we couldn't effectively fit sigmoidal models, clinal models to these shifts, but they were present across the transect. And we detected a steep shift in soil chemistry, which was an interesting result for me. This particular cline, I didn't have enough pollinator data, unfortunately, but the soil result was quite interesting. And if we look at the specific cline parameters, here I'm going to focus on what's called the cline center, which is the inflection point where the cline shifts along the landscape. And each of these black dots are specific outlier SNPs. And the colored dots represent, here, for example, soil chemistry, gene pool assignment, and multispectral phenotype. So there are different other non-outlier SNP variables, and each of these dots represent parameters in the cline. So what you can see is most of the outlier SNPs are situated on one contig in the assembly, so contig 24. So they're presumably very closely linked. But what you can also see is the cline center of the outlier SNPs and the whole genome signal does not correspond to the center of the multispectral phenotype. So the shifts are decoupled. So there's some signal for selection by soil, but it's very hard to determine because it's actually not linked to the morphological difference. And it's localized, it's highly localized in the genome. And we look at the tricolor Cardueae transition zone, there's similar patterns in shifts in genotype and phenotype and soil, and also no patterns in the pollinator communities. But importantly, in this condition, you can see there's a much higher number of outlier SNPs on a greater variety of contexts. And here, there's a correspondence between the transition in morphology, genetics, and soil regime. So different morphological variables correspond to different sets of outlier SNPs, and soil regime is corresponding to the whole genome change and some of the outlier SNPs. So certainly, here we have the strongest signal of selection along the lines. In contrast, in the final transition zone, the Cardueae citrus style transition zone, there's very few outlier SNPs. So you can see this, it's just localized to a few, and the gene pool assignment signal is lower. There is some shifts in morphology, but it's shallower. It's more shallowly linear, and there's no cline in selective regime. And very importantly, there's no correspondence between morphological shifts and the genetic shifts. So in fact, there's no signal of selection or allele surfing in this transition at all. So I thought that meant surely that the Cardueae citrus style transition would most likely be a candidate for IBD effects. And in fact, when we looked at the IBD patterns, IBD was strongest in the tricolor Cardueae transition, which also has the strongest signals of selection. So this is just a set of Mantel tests, which with the x-axis being geographic distance and the y-axis being genetic distance for each of the separate transition zones. And yeah, you can see the only significant result was in the transition zone with the highest signal of selection. And actually, if you think about it a little bit more, that may be an expected results, because on a transect you'd expect it to be some signals of IBD. And this transect also showed the greatest degree of genetic differentiation. So effectively, this is representing the whole genome signal, which is a sharp geographic shift. But it doesn't completely explain what's going on in the Cardueae citrus style transition, which remains a bit of a mystery. So in terms of take-home messages, signatures of selection can clearly be swamped in rapid radiations, but they are detectable if you use a full suite of data and methodology which have been developed over the past decades. So here they differ between the different transitions. So there's clearly multiple processes that interact to generate morphotypes. So selection in correspondence with drift effects, for example. And another important take-home I thought was that whatever morphological traits you pick for inferring the effects of selection can have a demonstrable effect on the actual outcome. So here we didn't measure soil-related traits such as root morphology because we presumed all the variation was floral. But in fact, we found strong signatures for selection from soil. So this may have produced an even stronger result for selection, which may have produced a more consistent taxonomic decision. And then lastly, recent diversification does make evolutionary independence of lineages very difficult to identify, but it's important to be transparent in the taxonomic decisions you choose and to consider evolutionary process case by case. Here, I was more conservative with my decision to elevate them to subspecies based on the fact that the transitions were not showing consistent evolutionary process. But I can definitely see the case where people may elect to use these subtle signatures of selection to elevate these entities to species level. And they're just perfectly justifiable, but it's important to be transparent in your decisions. So I just wanted to end off by speaking about, because this is the TICA forum, to speaking about some of my future work on Senecio. I'm currently working towards producing a target capture phylogeny of South African Senecio. And I just wanted to mention that because I know that the TICA community has a few members who work on Senecio, and it's quite an ambitious, large project that I have in mind. So I'm trying to work towards using the vast amount of target capture data which is being produced, producing a global phylogeny and reclassification of the tribe, and hopefully the genus Senecio. But that, people tell me, is going to be a lifelong endeavor. So I'm putting this out there so that I can foster future collaborations, because it's certainly not a job I can do alone. And I look forward to chatting to more people about it. So yeah, thanks everyone for listening to my talk. And I'd like to thank my supervisors for my PhD and all my many fieldwork and analytics assistants over the years. Thank you very much.
Thank you very much. So does anyone have any questions? I had one question about, so you have your nice transect with your different transitions between the four morphotypes. How complex, because there's obviously more than 4 morphotypes total. So are any of the other morphotypes also potentially interacting with these 4, or are they kind of 4 isolated ones and the other morphotypes are elsewhere?
No, so there is a situation on one of those transition zones where another morphotype comes in from the east, from one of the mountains. And if you include those populations in the overall population genetic analysis, you get the gene pool admixture. So there's definitely admixture from other different. I simplified this effective chapter into this one transect, but you could produce networks and produce more complex questions, where it obviously it asks a lot more of your brain to think of it in a multidimensional fashion. So I tried to simplify it, and I specifically looked at the endpoints of this transition for this question. But yeah, definitely, it's a very complex system, and certain morphotypes are much more clearly delineated than others, whereas others you get populations of where there's clear mixture, and others you get sharp grips. So I didn't look. Ideally, if I had all the resources and all the time in the world, I would have looked across all the transitions in the complex, but obviously that wasn't going to be possible. Yeah.
I was wondering about the pollinator community.
Yeah.
I'm assuming that you found some differences across the landscape. So can you speak just a little bit about it?
Yeah, sorry. So I was trying to keep strictly to the 15-minute time, and it was a large amount of data that I was sifting through. So I sort of skimmed over those details. But yeah, effectively, these things, the pollinator signals is very difficult because it's generalist pollinators. So they have basically a few that they're mostly fly pollinated, and they've got these knitted beetles, etc. So what we found was there were spikes in what I basically did was I did community observations, and I inspected 2,000 inflorescences per population, and recorded insect visitation on each of these populations, and then got frequencies. And what I found was there was spikes associated with certain zones, but the variation was definitely not clinal. So the clinal models didn't pick it up. So there is definitely some effects of pollinators, but it's super subtle and constantly shifting. So I don't exclude pollinators, but I just thought that it was interesting that the soil patterns were so much clearer. I think the pollinator patterns are there, but they're not clinal, so to say. Like so they're shifting on the landscape. So it's mostly this fly genus called Corsomiza, and another one which is a horsefly, the Regioglossa, that shows the most consistent patterns in the genus, in the system. Yeah. I don't know if that answers your question.
Yes, and one, I guess one last question. Are there any other morphological features beyond flower color and other more floral patterns that could separate each of your subspecies? I'm going more like seeds, for example, achenes or.
So the species I actually identified, which weren't in this talk, but the three species, they actually have quite distinctive differences in fruit morphology, in terms of the structure and the size of the fruits. So that actually makes them morphologically diagnosable. But the subspecies, the fruit, I haven't seen any major structural variation that would differ. The variation to my eye seems entirely floral. I did notice there's some difference in trichomes, but I haven't done extensive trichome slides of each of the subspecies to that extent, just as a virtue of time. And as I mentioned, I didn't measure root traits or anything like that. Certainly, it's very difficult to get leaf traits variation at a subspecies level in this kind of diversification, because they are annuals and they are very susceptible to differences in water regime and any disturbance. So the patterns get totally swamped out. You need like fast amounts of data, I think, to actually find subspecies specific leaf differences. But I think it's more likely that most of the variation is floral and some of the variation may be root, but I'm not so sure about that. Yeah.
Thank you so much.
Sure.
Well, thank you very much to all three of our speakers for a great series of talks today. And hopefully we will see you all in July for our next TICA Talks.
Thanks for organizing everything, Abby.